This is a spermatid, then that spermatid develops into a sperm cell. So we're talking about the same cell, spermatid to sperm cell, same thing, okay? Same individual, they just only happen to have to undergo development. And the sperm is stored in the epididymis until ejaculation. Then, so no pathways, I love pathways, and so passage of sperm, where does it go? Epididymis, vas deferens, ejaculatory duct, urethra. Okay, that's the pathway. It's a shorter pathway here. All right, so with spermatogenesis in the typical human male, about 3 million of them are produced a day. So it's once puberty is reached, it is a continuous process. Okay, still under hormonal control, but still a continuous process. If ejaculation does not take place, the sperm is stored in, there's a limited amount of space there in that tube. The body will eventually reabsorb all of that. Sperm cells will be broken down, digested, and new sperm cells will replace them. All right? Okay, now this is still under hormonal control. And ultimately, the control region is the hypothalamus. Okay? Now there's several different feedbacks here onto the hypothalamus, but first of all, before puberty, all of the sexual system is immature, non-functional, immature. Starting at puberty, I'm not really sure about what triggers it, but the hypothalamus will start secreting gonadotropin releasing hormone. Okay, gonadotropin releasing hormone. I mentioned before that one of the ways that the anterior pituitary is controlled by the hypothalamus is through the release of releasing hormones or inhibitory hormones, okay, that travel a very short portal system to the anterior pituitary and will bind to the appropriate cells. Okay, in this case, there's an increase in gonadotropin releasing hormones. The target is the anterior pituitary, and not just anything in the anterior pituitary, but only those cells that will produce two different types of hormones. And these are not steroid hormones, they're short peptides. Okay, so this effect, so the target, gonadotropin releasing hormone, anterior pituitary, cells within the anterior pituitary, and this stimulates them to release two different hormones. Follicle stimulating hormone. Even though males don't have follicles, this hormone was first discovered in females, but both males and females have them. And but their functions slightly different. And luteinizing hormone, okay? You just need to know these as by the acronyms. I prefer to say follicle stimulating hormone or luteinizing hormone because it's easier for me. I hate acronyms. All right, so nonetheless, you can just mention these. The target of these are the cells in the male reproductive system. They're immature at this time, and this stimulates the maturation of the reproductive system. It also targets our other tissues in the body that lead to secondary sexual characteristics. Now, what are secondary sexual characteristics? Is that they are characteristics that we find differences between males and females, but those differences are not directly related to reproduction or to the reproductive system. So the direct control here is maturation of the reproductive system. But other things happen as well. Facial hair and the growth of hair in other parts of the body, that is stimulated. Also the increase in muscle mass, okay, that is a secondary sexual characteristic. Furthermore, deepening of the voice. That is also something that happens in males as well. And so these are secondary sexual characteristics, and they are induced by these two hormones, luteinizing hormone, follicle stimulating hormone. Now, once sexual maturity has been reached at puberty, these same two hormones and the hypothalamus, they still control the rate and process of spermatogenesis. And usually it's a pretty steady state. It doesn't change all that much. Okay, so the hypothalamus still stimulates the anterior pituitary by releasing gonadotropin releasing hormone. Okay, that's still necessary, gonadotropin releasing hormone. And the pituitary will release into the bloodstream luteinizing hormone, follicle-stimulating hormone. And of course, the targets are in the gonads, in the testes. But that's too broad. It's very specific cells, okay? And so luteinizing hormone, their receptors are found on cells between seminiferous tubules, okay? I mentioned with seminiferous tubules inside, there are Sertoli cells. But now here's a different type of cell called Leydig cells, and they are in the regions between seminiferous tubules. Okay, these are the androgen-producing cells. Androgens are a collection of so-called male hormones, but of which testosterone is the primary one. Okay, there are others, and we're not going to talk about them, but testosterone is the major one. These are steroids, okay, that derive from cholesterol, ultimately. Okay, and although these were called androgens, females have them too. Okay, and in fact, the so-called male hormones, female hormones, both males and females have them, just in different proportions. That's all. Okay, so this here mostly testosterone. All right, so the target of testosterone are Sertoli cells. Okay, these are the nerve cells that are within the seminiferous tubules. They have connections, cytoplasmic connections to spermatogonia, primary spermatocytes, secondary spermatocytes, and everything. This is also a target of follicle-stimulating hormone. Okay, so both of these hormones will bind to their separate receptors on Sertoli cells, and this here leads to spermatogenesis. Okay, induces spermatogenesis, meiosis in the seminiferous tubules. Now there's also another target for the androgens. It's negative. You might not see that line there, but there's a line, and it is feedback onto the hypothalamus. And so this is actually a negative pathway because high androgen levels, it has an inhibitory effect on the hypothalamus. So the hypothalamus would lower its levels of gonadotrophic releasing hormone. The effect would be then that the anterior pituitary, with the absence of that stimulation, their secretion of luteinizing hormone, follicle-stimulating hormone, would also decrease. Okay? Once that decrease goes down far enough, however, that inhibition is released and we get the system going again. Okay, so now this negative feedback here and this positive feedback that way keeps the system pretty much in equilibrium. Okay, it's not like it's going up tremendously and going down tremendously. It stays in pretty much a steady state because of the two legs of this particular thing. There's no cycles here, all right? And so spermatogenesis is something that is going on continuously. All right, so that's the hormonal control of the male system. You're going to love the hormonal control of the female system in mammals. Okay, so we'll get into that. So the female reproductive system, egg reproduction, this is called oogenesis. So in males, production of sperm is called spermatogenesis. This is oogenesis. Anytime you see this genesis, it means to generate. Okay, so this is system to produce ova, the technical scientific word for the gamete that we've been calling eggs, right? Okay, it's much more complicated than in a male, and I'm talking in general about mammalian systems here. We're not going to worry about other types of vertebrates, just only mammals. Most mammals, other than humans, they go through estrus cycles. Humans do not have estrus cycles. So what's the difference here? Is that sexual receptivity by the female and also breeding, because the female allows males to breed, okay, is that it's seasonal. Okay, it's not continuous. Seasonal. There's very distinct times of the year when the female is sexually receptive. And it coincides with the production of ova, with eggs. All right? Furthermore, the lining of the uterus, Okay, I'll get to the anatomy here in a moment, but the lining of the uterus is a special muscular tissue called the endometrium, okay? This is still part of the uterus, but what is significant about the uterus is this is where any fetus will become embedded. This is where the fetus will grow, all right? Is the part called the uterus. And in preparation for pregnancy, this here builds up, okay? It builds up a big, rich blood supply and a bunch of other tissues there because this is where an embryo will implant into the wall of the endometrium. Now, if no fertilization takes place, so this does build up, but if no fertilization takes place, then it will be broken down and all of the products absorbed by the body, okay? And, but humans are different, okay? We don't have estrous cycles. We have what is called menstrual cycle. And so there's cyclic changes in both the uterus and in the ovary, okay? So with this, female sexual receptivity is not seasonally limited. And furthermore, it doesn't coincide with the most likely time to become pregnant either, all right? That's with estrous cycles, there is both their behavior, female behavior, and the presence of ova for fertilization, they coincide. But here, there is no coincidence whatsoever. Okay, that's one thing. Another difference is that if fertilization does not take place, the endometrium is not reabsorbed. It is simply broken down. And so there is a loss of blood, a loss of tissue. And this is the monthly, about once a month. It's usually a cycle of 28 to 29 days. But nonetheless, the menstrual period is the loss of the lining of the endometrium. Then it will begin in the next cycle to build up again, always in preparation for the implantation of an embryo. And if fertilization does not take place, then once again, there will be a menstrual flow. Okay, now this also involves monthly changes in the ovary. So there's two things that happen here. There's an ovarian cycle that cycles on every about, as I said, 28, 29 days, and also a uterine cycle, changes in the endometrium lining of the uterus. That goes through this monthly cycling as well. And all this is under hormonal control. So as far as the anatomy, externally, the clitoris, it's just at the anterior part in front of the vagina. This is developmentally homologous to the penis, okay? So in early embryos, they're neither male nor female. When they develop, however, if it's under stimulation to become a male, that tissue will become a penis. If it's not stimulated to be a male, to be a female, then it becomes the clitoris. I'll answer your question here in a moment. On each side of the vagina is also flaps of skin called the labia minora, labia majora. They protect that area, but they are developmentally homologous to the scrotum in males, okay? So they develop from the same embryological tissue. Yes?
I have two questions. So first, for the endometrium, is there a reason that we shed it instead of absorbing it?
That's a good question, and no one has a good answer for it. Yeah, that's one of the things. People have come up with this type of hypothesis, this type of hypothesis. Many of those hypotheses are absolutely not even testable, and so it's really not known. It is a curiosity that humans, because our closest living relatives, chimpanzees, they have estrous cycles. Okay, it's only humans that do this, and why? No one really knows. There's no real answer for it. Okay. Okay. Now internally, the opening here is the vagina, and that is what receives the penis during copulation. Beyond that is the uterus, and there is a division between the uterus and the vagina called the cervix. There's an opening there, and this here will change in monthly as well, but I'm not going to talk about changes in that particular part of the cycle. Then on each side, so there's just one uterus, one vagina, but on each side coming off of the uterus is fallopian tubes. Okay, these are also called oviduct. Those are homologous, same word, oviduct or fallopian tubes. You hear them both. At the end of the fallopian tube, or the oviduct, I'm going to prefer oviduct, this funnels out, so there's an opening, so funnel shape there, and then there is the uterus, or excuse me, the ovary. The ovary and the oviduct are not physically connected. Okay, there's a space between them. Now, the ovaries are hanging from tissue that keeps them in place, but the oviduct sort of partly goes around one part of the ovary, but there's a fluid-filled space between them. They are not in physical contact. Okay? And so these are the parts that I'd like for you to know. All right? Okay, so looking at it from the anterior end, then there's the vagina, cervix, uterus. Endometrium is the lining of the uterus. Okay, this is usually very muscular, smooth muscle, but here in the lining of it that will build up and break down is called the endometrium. Okay, then we have the oviduct. This is where fertilization takes place. Okay, not down here, but way up here. All right, and then each side there are the ovaries. Okay, so these are the piping in the female system. Oogenesis, okay, the production of, actually brings about not only the production ultimately of an ovum, eggs, but of follicles. Okay, like you saw in lab yesterday, what is a follicle? Follicle is an oocyte surrounded by nerve cells called follicle cells. All right? Now, this here, oogenesis in females, begins before they're ever born. Okay, in the fetus. In the fetus. The beginning of meiosis will begin in the fetus, in the developing ovary. However, so it starts out with, first of all, an oogonium, diploid oogonium. Oogonium will then divide and develop into primary oocyte, still diploid. And then that primary oocyte will start meiosis 1 and go only to prophase 1 and stop. And it stays that way until puberty. Okay? So what are the events that happen in prophase 1 that makes it different from mitotic prophase and from prophase 2 of meiosis? What events happen in prophase 1? Crossing over, but first of all, before you have crossing over, in order for crossing over to happen, it must happen. You're right, you're right. Okay, but there's something that has to happen in order for crossing over to occur. What is it? What crosses over?
Lining up of homologous chromosomes.
Homologous chromosomes. So synapses, okay? The pairing of homologous chromosomes, okay? The two chromosome number 1s come together. Two chromosome number 2s come together. Two chromosome number 3s come together. Two X chromosomes, they synapse, all right? And we start crossing over, but they stop and stay that way. Then female, and in fact, all of the possible follicles that the female will ever have are produced before she's born. That's quite different than in males. All right, so this oocyte is a primary oocyte, and it's surrounded by nerve cells that are called follicle cells. So the whole structure is called a follicle. So these are primary follicles, and a primary follicle has a primary oocyte and one row, one layer of follicle cells. Okay, and it stays this way until puberty. Okay, so the female has been born, All the follicles that she'll ever produce in her life are there. And once the hormones generated, not all of the follicles will develop. Just only a certain class of a certain group of follicles every month. Well, actually, even before that, maybe about 6 months before they will ever go into a cycle, they are induced by follicle-stimulating hormone to develop further. And they will develop into a secondary oocyte, and then into, or secondary follicle, not oocyte, secondary follicle, then a mature follicle. By the time then it's going to enter into the monthly cycle, there will be about 20 of them, only 20, only one of which will ever survive, okay? So there's a lot of loss as we go through here, okay? So once it starts, it's been recruited into the monthly cycle, that particular month, is that it will eventually, that primary oocyte will eventually complete meiosis 1. Cytokinesis will be very unequal. We will produce what is called a secondary oocyte, one that has a lot of cytoplasm, and a cell that has very little cytoplasm, but still has a full set of haploid set of chromosomes, called the first polar body. That will be non-functional. Usually it will decay, be reabsorbed. So we only have the secondary oocyte that will proceed to metaphase 2 and stop. Okay, so there's 2 stops here. First, at prophase 1, from before the female is ever born up until it enters into a monthly cycle. During the monthly cycle, during that time, it will complete meiosis 1, start meiosis 2, and stop at metaphase. Okay, it won't go any further. All right, now I'm going to not impose upon you this next 2 slides. I want to just run through just briefly. This is what I want you to know. You don't have to know all this stuff. Normally I do talk about it, but I want to move on. So first of all, in the very beginning, the female would produce about 7 million, eventually 7 million follicles. And as we go through all of this process up to when she becomes reproductively functional, a lot of these follicles are just lost. Okay, there's very few of them ever lost. For ovulation, within her lifetime, and within her reproductive lifetime, there will be about 450 to 500 actual follicles that ever become ovulated. So most of the follicles will be lost, all right? And you don't need to worry about this either. I'm just wanting to point out, the only thing I want to point out here is, yes, everything, the oocyte, primary oocyte, has been in prophase 1. Once she reaches sexual maturity, puberty, then maybe about 6 months before the actual period, the cycle for which this group of cells, follicles, are going to be involved, some of these primary follicles will be induced to develop over a 6-month period. And then the next month, another group will be induced. And then over the next month, another group. So they're in waves. However, when we get to the end, starting the first monthly cycle, there will only be about 15 to 20 large, mature follicles that will be induced by hormones at the beginning of that monthly cycle. But I want you, so what is the take-home message here? Is that these follicles start developing from primordial follicles a good 6 months before they enter any ovarian cycle, the monthly cycle. It takes them that long, and a lot of them are lost along the way. Okay, they just don't develop, right? And by the time you reach the beginning of the cycle, there will be about 15 of them that can enter and will be recruited, right? So that's the only thing I want you to know about this. So what will happen now, with all these follicles, don't worry about androl. All these follicles are really dependent on follicle-stimulating hormone. And the receptors for the follicle-stimulating hormone are not on the oocyte. They are on the follicle cells, okay? The follicle cells, just like the Sertoli cells in males, have cytoplasmic connections to the oocyte, and they move materials into the oocyte. In fact, they're controlling a lot of the oocyte. All right? So the follicle cells at this time start producing a hormone called estrogen. Okay, so the oocyte doesn't do it. The actual source of estrogen is the follicle cells. And they're induced to produce this by levels of follicle-stimulating hormone. Okay, follicle-stimulating hormone. These are the cells that have the receptors for follicle-stimulating hormone. At this time, even though the anterior pituitary is putting out luteinizing hormone, there are no receptors for luteinizing hormone here. Okay, so it doesn't do anything. Not yet. So as we enter into the monthly cycle, there will be a slight increase in follicle-stimulating hormone. This slight increase then stimulates what is called the follicular phase of the ovary. So this leads to about 15 to 20 mature follicles are said to be recruited. Okay, they're going to develop yet further. All right? However, in humans, now in other animals, there'll be several of these will make it and become fertilized. Okay, they have litters, like rats and cats and dogs. But in humans, normally, out of these 15 to 20, only one of them will survive. The rest of them will disintegrate. And which ones survive are the ones that have that are in follicles that have a large number of receptors for follicle-stimulating hormone. So the winning follicle is the one where there's an abundance of follicle-stimulating hormone receptors. Therefore, the message gets to be very high on these. Okay? All right, so we have usually one dominant follicle. There can be sometimes two, in which case, if they both, when we get ovulation and they both get fertilized, you have fraternal twins. Okay, not identical twins, fraternal twins. So this is also called dizygotic, meaning that they came from two separate zygotes. Identical twins, however, come from one zygote. One zygote that has developed into at least an 8-stage blastula, not even blastula, morula, and if one of those cells gets separated, it still develops into an identical individual. All right? Okay, so this is still the primary oocyte at the beginning of this monthly cycle is still in prophase 1. Still in prophase 1. Once we enter the cycle, about day 14, that's about halfway through, then there's a large surge of luteinizing hormone. And just prior to reaching this point, those follicle cells have now developed receptors for luteinizing hormone. Up to now, they didn't have. They could only respond to follicle-stimulating hormone. But now they can respond to luteinizing hormone. And so this primary oocyte, on that surge, that is what triggers it to complete meiosis 1. Up to that time, it was a primary oocyte. So it quickly completes meiosis 1, resulting in a secondary oocyte and the first polar body. It will proceed into meiosis 2, but stop at metaphase. And so it will remain a secondary oocyte upon ovulation, as I'll get to here in a moment. So unequal cytokinesis, first polar body, not functional, and then secondary oocyte. It is in, and it's haploid. It's haploid. This is where the reduction in chromosome set numbers has taken place, and it stops at metaphase 2, and it remains that way. So what is ovulated? What does ovulation mean? Okay, so something, I mentioned this about day 14, and it's variable. This is just an average date, okay? About 14th day, we have a surge, peak, and luteinizing hormone. That triggers that primary oocyte to complete meiosis 1, start meiosis 2, but also after that, you have ovulation. So this surviving mature follicle is real big, near the surface, and what happens with ovulation is that the wall of that follicle ruptures, and it releases into the body fluids near the ovary. It releases the secondary oocyte, along with a cluster of follicle cells. So what is ovulated is not an egg. It's not an ovum. It's a secondary oocyte, okay? Secondary oocyte, that's what is released, okay? So now this, as I said, the oviduct is not physically connected, but it has a funnel shape to it. There are cilia inside of the cells lining that oviduct. They are beating and they're drawing fluid down into the oviduct. And so usually the secondary oocyte plus its follicle cells are drawn into the oviduct, okay? And then it starts its journey down the oviduct. All right, so this is what is really ovulated. This is called ovulation. Now, the follicle cells that remain back in the ruptured follicle will now develop into something that is called the corpus luteum, okay? So these developed from follicle cells, okay? It's really big. And this will start secreting another steroid hormone, progesterone. Okay, so the follicle cells are the source of 1, estrogen, and after ovulation, 2, progesterone. All right? They produce both of these. Not the primary oocyte, not the secondary oocyte. Those are not producing any hormones. It is the follicle cells. Once they have ovulated, this is now turned, the remaining follicle turns into corpus luteum. All right, so this is sort of showing the cycle. First of all, primordial follicle, eventually grows, and I'm not worried about antrum. You don't have to worry about that. But by the time it gets to a mature follicle, it is a secondary oocyte near the surface, very large follicle that ruptures, and it is the secondary oocyte that is ovulated. The follicle that's left behind becomes corpus luteum. However, the corpus luteum is dependent on luteinizing hormone for its existence. So in order for that corpus luteum to remain, it has to have stimulation, continuous stimulation with luteinizing hormone. Luteinizing hormone, very important here. It is producing progesterone. Progesterone and estrogen during the cycle. First of all, estrogen. It has stimulated cells in the endometrium to start vascularizing. So we're starting to build up a network of capillaries and arteries, blood vessels into the wall of the endometrium. After ovulation, in order for that endometrium to remain, it requires progesterone. If progesterone levels disappear, this here then triggers the breakdown of the endometrium, as I'll get to here in a moment. Now, where fertilization will take place is in the oviduct. And so sperm that is deposited in the vagina, it has to get through the cervix, into the uterus, and even beyond. It has to get up into the oviduct. And so that is a long ways for a sperm cell to swim. And so the idea that that sperm is moved up the system simply by the beating of the flagella, it is in part, but it is also what I mentioned before, that in the male semen, there are prostaglandins which stimulate rhythmic contractions of the smooth muscle in the uterus, which basically squeezes this stuff further up into the oviduct. Okay, and it can store sperm. Sperm can live for about 5 days in the system. After that, you don't have to worry about it, it's gone. So if intercourse takes place up to 5 days before ovulation, it's still possible for the female to become pregnant. Okay. All right, so this here, what the sperm cell will meet, is not an ovum. The sperm cell comes in contact, it comes in contact with a secondary oocyte. It is a secondary oocyte that has been ovulated. All right, so the sperm cell will make contact. There are receptors, species-specific protein receptors, both on the sperm cell and on the oocyte. When it makes contact, this leads to a depolarization. Okay, so the plasma membrane of the oocyte will undergo a electrical reversal. All cells in the body have positive charge on the outside and negative charge on the inside. This here reverses, okay, shortly. And that is what triggers the completion of meiosis 1, or meiosis 2. So, first of all, the sperm cell has to make contact, then the nucleus of the secondary oocyte rapidly completes meiosis 2, resulting in an ovum and a secondary polar body. By this time, the sperm nucleus is already in the cytoplasm. So now you have an ovum nucleus, sperm nucleus, then they fuse, and it becomes a zygote. Okay? So those are the events that happen here. So this will complete unequal meiosis 2. We talked about the secondary oocyte, second polar body, and an ovum, which we call also an egg. All right? So this sperm nucleus, ovum nucleus will fuse together, and now you have a diploid zygote. Okay? Any questions? Okay, this is the chain of events that happens. All right, so this zygote will then begin mitosis. And here, all of the nutrients for energy and also for materials to build structure are already there in the cytoplasm of the ovum. That's why the ovum as a cell is really big, okay? Much bigger than in the sperm cell. So this will start dividing. It is not feeding, okay? Might take up a little bit of stuff from secretions in the oviduct, but primarily it's using its supplies of yolk that happen to be stored in the original oocyte. So it'll divide once, divide again into a morula, and then a blastula. In humans or in mammals, this blastula stage is called a blastocyst, okay? And although in most animals, as I pointed out before, the blastula is a hollow ball of cells, one layer thick, the blastocyst is a little bit different because it does have a blastocele, fluid-filled space on the inside. It is mostly one layer thick, except for on one end, it has something that's called the inner cell mass. Now, this here, inner cell mass, is what will become the embryo, the individual. All of these other cells out here will not be part of the embryo. They will produce the extra embryonic membranes. Okay? So this is true of all amniotes. Okay, not just the mammals, all amniotes. All right? So the important point here is that these cells that will become the chorion, allantois, and so forth, they all happen to be genetically identical to the cells that will become the embryo. They are genetically alike. They came from one fertilization, one zygote. Okay, but not all of the zygote progeny cells will become a human or any other animal. Okay, so fertilization is taken up here, and so it takes some time for this to get down. It does have some secretions of the uterus that supply it with material, but also a lot of that material was stored as yolk. And it's moving by fluids. It can't swim or anything, but it's being brought down by the flow of fluids that is being propelled by ciliated cells that line the oviduct. All right? And it goes toward the uterus, and about 6 days, plus or minus, these are averages, then it will reach the uterus. By that, and the endometrium, of course, has highly vascularized by that time. When I say vascularization, we're talking about blood supply. It is very enriched with a lot of blood supply, and then this will embed as a blastocyst. Okay, it's a blastocyst at this time. All right? And now, what happens if no fertilization takes place? Meiosis 2 will never be completed. It will never develop into an ovum. It will stay a secondary oocyte, and it will decay. It and the follicle cells associated with it will simply be sloughed off with the sloughing off of the cells of the endometrium during the menstrual period. So it requires fertilization, or at least contact, not fertilization. It requires contact with a sperm cell to induce the formation of the ovum. Okay, so this will just degrade and it'll be lost in the menstrual flow. Okay, all right. Now, the hormonal controls on this. This is what gets really interesting. Still hypothalamus, still the same players, except for here we have estrogen and progesterone that now come into play. Males also produce progesterone, and it's not really understood what role it plays in males, but it's pretty well understood how it works in females. So still the hypothalamus is putting out gonadotropin releasing hormone, which then has to stimulate the anterior pituitary to release follicle stimulating hormone and luteinizing hormone. And of course the targets, keep in mind, for all these hormones that I've been talking about, I always want you to know their source. I want you to know what triggers their release, the stimulus, and what their targets are, and what the response of their targets. And if I mention very specific cells or tissue, that is the target. Okay, you can't be broader. Like with the kidneys, the target for antidiuretic hormone is not kidneys. That's too broad. Okay, it has to be collecting ducts. Right? So here, follicle stimulating hormone, the target is follicle cells. Okay, follicle cells. Also for luteinizing hormone, follicle cells, but not at the same time, because the follicle cells always are responding to follicle stimulating hormone, but only late in the monthly cycle, just before that surge in luteinizing hormone, about day 14, they respond to luteinizing hormone because they have the receptors for it. Okay? Other hormones here produced by the follicle cells in the ovary. I have ovary here, but really the follicle cells. Okay, this is the source for estrogen and progesterone. After the follicle ruptures, becomes corpus luteum, then it will start secreting, it's the corpus luteum that is the source of progesterone. Okay, so now this next slide here is going through the monthly cycle, and in these columns that I put here, they're showing the events happening in different parts of the body. So this first column is referring to the hypothalamus. The second column to the anterior pituitary. Third column to events that's happening in the ovary. And the last column is events that will be happening in the uterus. Okay, because all these are involved. And I'm going to start at the beginning of the cycle, okay? The very beginning, just right after the last ending of the last menstrual flow. Okay, so we're starting a new cycle. At this time, the hypothalamus is putting out very low levels of gonadotropin releasing hormone, which then is stimulating the pituitary to put out low levels of follicle stimulating hormone. Now, even though I'm doing this in discrete steps, I'm going to be walking through discrete steps here. In actuality, this is a smooth continuum. Things gradually change. This is called, the first part of the cycle is called the proliferative phase, okay? And so the targets at this time in the ovary are the follicle cells, okay? Now, at this time, they cannot respond to luteinizing hormone. They do not have receptors for luteinizing hormone. They only have receptors for follicle-stimulating hormone. And so this is starting to recruit those 15 to 20 large follicles, right? So at this time, under the stimulation of follicle-stimulating hormone, the follicle cells start secreting estrogen. Okay, estrogen is a steroid, and therefore its effect, remember that steroids like this would bind to an internal receptor, become a transcription factor. So this is affecting development and cell behavior, okay, with gene expression. So this is still low levels, but the target of estrogen is cells in the endometrium, the lining of the uterus. And once we have low stimulation, it then stimulates growth. Now, I say here rapid cell division and vascularization. This isn't so rapid right now, okay? But still, it's higher level mitosis than what you see in most other tissues of the body. In other words, the wall of the endometrium begins to build up, okay? Begins to build up, all right? Okay, now, estrogen also has two targets, okay? One is the cells in the endometrium, and so this is encouraging the development of a highly vascular, will eventually be very vascularized, but also the hypothalamus. This estrogen at low levels is stimulatory. And so with the stimulation of the hypothalamus, then the levels of gonadotrophin releasing hormones go up. So as estrogen levels rise, it stimulates the hypothalamus to increase its stimulation of the anterior pituitary. So these levels go up, gonadotrophin releasing hormone, which means greater stimulation on the pituitary gland, which means that the release of higher amounts of follicle-stimulating and luteinizing hormone. Okay, so unlike in the male, those levels stay relatively the same due to the feedback, but this here is a positive feedback which is increasing these. And of course, what is the target of follicle-stimulating hormone is the follicle cells. So as that goes up, then the follicle cells are more stimulated, and therefore we get more growth in the follicle, and as a consequence, estrogen levels also start going up. Because this is all a positive feedback loop. And I'll jump to the next screen. We're going to continue. So as these estrogen levels increase, then that gives even greater stimulus on the endometrium to form. So these are all rising with positive feedback. And of course, estrogen, its second target, is the hypothalamus. So by getting, now we're getting into the ovulation phase. And so we're about day 13, 14 of the cycle. So all these hormones have been gradually going up due to positive feedback. We get high levels of when estrogen reaches the high enough levels, then if we have real strong stimulation on the hypothalamus, and there is a sudden high peak increase in both follicle-stimulating hormone and luteinizing hormone. So this is the LH surge that I was talking about earlier that happens, that stimulates one, the follicle cells to induce the primary oocyte to complete meiosis and start meiosis 2. All right, now by this time too, the follicle cells have receptors for luteinizing hormone. And therefore, ovulation is triggered. Ovulation is triggered by the follicle's response to luteinizing hormone. So luteinizing hormone goes up, and within And, you know, this is an average, it could be 24, it could be as much as 36 hours after this surge, then the follicle ruptures and we get ovulation. So what triggers the ovulation is a response to the high levels of luteinizing hormone. Okay? What's left behind after ovulation becomes the corpus luteum, which secretes progesterone. Everyone have that? Now, the secretory phase is after ovulation. And so the corpus luteum is secreting higher levels of estrogen and also progesterone. Okay, which both of them stimulate the endometrium and it reaches its highest level of vascularization. Okay, it's fully developed. Now, the maintenance of this endometrium at this time is not so much maintained by estrogen. It is progesterone. Okay, this is the major hormone which is keeping the endometrium intact. All right, that's a real important point. I'm going to state, it's so important that I'm going to state it again, that what maintains the high level of vascularization and development of the endometrium at this stage is progesterone. Okay? All right, now we have two hormones in high amount. Once again, both of them, so there's two targets. I already mentioned for estrogen, it is the endometrium is one target, the hypothalamus is the other. Progesterone, the same thing, okay? The endometrium, but also the hypothalamus, but now high levels of progesterone and high levels of estrogen are inhibitory. Estrogen by itself was stimulatory. The more estrogen, the more stimulation we had on the hypothalamus. But now with progesterone there, high levels of progesterone, then levels of, this is inhibitory, and levels of gonadotrophin releasing hormone drop. Well, the gonadotrophin releasing hormone was necessary to stimulate the anterior pituitary. Therefore, levels of follicle stimulating hormone, luteinizing hormone, go down. They start to decrease. And as our levels of luteinizing hormone drops, well, the luteinizing hormone is what maintains the corpus luteum. Luteinizing hormone maintains the corpus luteum. So as LH drops, the corpus luteum starts to degrade. But what does the corpus luteum produce? What does the corpus luteum produce?
Progesterone.
Estrogen and
Progesterone.
progesterone. Progesterone is real important here because that's what maintains, at this stage, maintains the endometrium. So we're now, that's starting to decay, and as a result, levels of estrogen, progesterone are dropping. And therefore, when progesterone reaches a certain low threshold, it leads arteries that are in the endometrium to spasm and cut off the blood supply, killing the cells in the endometrium. And the cells die, and the capillaries and cells will slough off. This is the beginning of the menstrual flow. Now, what inhibited the hypothalamus was high levels primarily of progesterone. But as the corpus luteum in these last 13 or so days decays, then progesterone is dropping, and that will then remove the stimulus on the hypothalamus. And it starts secreting low levels of gonadotrophin releasing hormone. And now we're back to where I started. We've gone through one whole month. We're now back at the beginning, right after the menstrual flow. So this is the normal controls on it. And as I said, it's a continuum. I've done this in steps, but it's really continuum. So to see this menstrual phase, which is the last part of the cycle, lasting in most humans about 3 to 5 days, okay? Is that there's a lot of things that happen. There's enzymes that are called metalloproteases, that they are released by the cells, and this breaks up the extracellular matrix. Remember that the extracellular matrix is a combination of various types of glycoproteins, collagen, that are outside of the cells that hold connective tissues together. Therefore, that fails, and this is what's leading to all those dead capillaries, dead cells sloughing off in a blood flow. Furthermore, there's types of arteries there that are called spiral. Those arteries, they contract, and this is what's breaking down, preventing the blood flow into the endometrium, and you get what is called hypoxic necrosis. Necrosis means death, okay? So death of cells, they die because they don't have oxygen. Okay, this word hypoxic means low oxygen, and therefore they, because of the blood supply. So the outer 2/3 of the endometrium will then break down. Furthermore, there's inflammatory prostaglandins. I mentioned before, prostaglandins, these are fatty acid signals that derive from a 22-carbon fatty acid that I mentioned before. You don't need to know the name of it. These were also produced in males and deposited with semen in the females, and the function of those prostaglandins from the male was to stimulate muscle contractions in the female uterus. Well, these also do similarly. However, this is what leads to contraction of the smooth muscles that are pushing the cells and material out, and this here contributes to the cramps that some females will experience during this time, is due to these prostaglandins. Okay, so the outer 2/3 of the endometrium will be lost, and usually that totally accounts for about 25 to 35 milliliters of blood and material. All right, so that's what happens now. So yeah, let's go back here. You might get yourself familiar a little bit with this. What I just covered in those last few slides were step-by-step certain phases. As I said, they happen to be somewhat of a continuum. So if you look up here, this is the levels of hormones. I mentioned that there was a little, at the very beginning of the cycle, there was a little bit of a surge in follicle-stimulating hormone, and that's shown here. Then it drops, but it's this little surge right here which recruits about 15 to 20 mature follicles to start into the cycle, only one of which normally will survive. Mid-cycle, we have this sudden surge in both follicle-stimulating hormone, luteinizing hormone. That is what triggers ovulation. These are the changes that are happening during this time in the ovary. A follicle will grow, become mature. We get ovulation, coincides with that peak in luteinizing hormone, and then corpus luteum, but this peak, or this will start producing progesterone. So we see that progesterone happens after that because it's from the corpus luteum. But as we get high progesterone at this point, it will stimulate or negatively inhibit the hypothalamus, and everything goes down, this decays, and this is what's happening in the uterus. Builds up, and then once we no longer have high levels of progesterone to maintain it, it'll lead into the menstrual flow. Then we're back at the beginning, and this happens in a cyclic fashion. Okay? All right. Now, if, however, a blastocyst invents itself in the endometrium, we have a problem, don't we? We don't want the endometrium to break down. If the endometrium breaks down now, the baby is lost. Okay? What maintains the endometrium? What hormone maintains the endometrium? Progesterone. But we just had high levels of progesterone, and our luteinizing hormones going down. Yeah.
On our slide,
right? It says 12 days after fertilization, but yours only
It
says,
says what?
ours says 12 days after fertilization, and yours only says 67 days.
Yeah, well, this is, go to this. I'll have to change what I have there in the notes. What is it? What do I say? It'll take 12 days, yeah. It's really, well, that 12 days is probably after, pretty close to after the LH peak, the surge. But still, take it out, this is after, okay? I'll have to fix that, all right? So, now, you see a problem here? Because after we have, the corpus luteum is breaking down, because luteinizing hormone is going down. But if that corpus luteum breaks down, then our progesterone is going to disappear, and we're going to lose this fetus, this black, because the endometrium will slough off. So really what happens is that the embryo, once it embeds, it starts secreting another hormone called human chorionic gonadotropin. And it is like LH. Its target is the corpus luteum. Our levels of LH do indeed drop and disappear. Okay, they do. But this is what maintains the corpus luteum. So human chorionic gonadotropin is maintaining the corpus luteum. Corpus luteum is still putting out high levels of progesterone, and the high levels of progesterone maintain the endometrium, which we want. Otherwise, the fetus is gone. All right? All right, so this here maintains the endometrium. Now, this is during pregnancy. In fact, it's the presence in the female's blood of human chorionic gonadotropin that is the basis of pregnancy tests. So these tests, it gets into the urine because this is being produced at high levels by the fetus that's embedded. It gets into the female's blood, gets into the kidneys, gets into the urine. And so the urine test for pregnancy, they're checking for the presence of this hormone. There is no way that you would have a positive test for this hormone if the female was not pregnant, because the female does not produce this, the fetus does. Okay, so that is the basis of what's going on here. Okay, so eventually a placenta will form by day 45. This after, it's counting these days from fertilization. Okay, counting the days from there. All right, and this is a combination. The placenta is a complicated thing. Not going to go into its structure, but there are both maternal tissues and fetal tissues that are involved here. So allantois and not so much chorion are in, part of the chorion, and allantois are part of the tissues that are in the placenta. And the whole point here is that the two blood systems, the circulatory system of the developing embryo and mother's blood, do not mix because they have incompatible proteins. And if they mixed, the mother's immune system would make antibodies to baby proteins and kill it. Okay, so they have to stay apart. And so there's capillary beds in here where you still get exchange of nutrients and waste products between the mother and the child through the placenta, but their blood supplies do not mix. Okay? All right. Now, eventually, by the second trimester, it's the placenta that begins to secrete progesterone. All right? And then the embryo ceases secreting the human chorionic gonadotropin. And so, since it's the source of progesterone is now coming from the placenta, you don't have human chorionic gonadotropin that would support the corpus luteum, and the corpus luteum decays and disappears. But the endometrium is still maintained, because now we still have progesterone. Now tell me, during pregnancy, is it possible for a female to get pregnant again? during that time? Is it possible to have another pregnancy during the 9-month period it takes the baby to develop before birth? Okay, explain to me. Some people say no. Explain to me why.
They're not releasing any eggs.
What's that?
They don't release any eggs.
Too far away, masking away.
They don't release any eggs.
Why not?
Because they're maintaining the baby.
I have to have a good reason, a good biological
I
reason.
don't remember the exact name, but I think that a mucous membrane forms on the cervix that would prevent any sperm from entering it.
Though that happens every month, actually, so that's not it. I already told you. I've already explained. You should be able to put two and two together. Why? Based on hormones. In order to enter the monthly cycle, what is necessary? Let's go back to the very beginning of the cycle. What has happened in the hypothalamus? It has to release gonadotrophin releasing hormones, right? Which would then lead to follicle stimulating hormones, luteinizing hormones, and get into the whole cycle. By the time you get to the second phase, the secretory phase, what hormones are high? After ovulation, what hormones are high?
Estrogen, progesterone.
Progesterone. And estrogen, but mostly progesterone. And high levels of progesterone do what?
It inhibits the
They
hypothalamus.
inhibit the hypothalamus. High progesterone inhibits the hypothalamus from releasing gonadotrophin releasing hormone. Follicle stimulating luteinizing hormone remain low. We still, all through here, have been maintaining high levels of progesterone. All through the pregnancy, there's been high levels of progesterone. Therefore, that has inhibited the hypothalamus from starting any new cycle. Incidentally, birth control pills have an artificial form of progesterone. That's how they work. By keeping those levels of this hormone high, it prevents any sort of cycle whatsoever because it suppresses the hypothalamus. Now, some of the birth control pills, it's called progestin. It's an artificial form, synthetic form of progesterone, but still works just fine. Sometimes some combinations will also have estrogen in it. And so there's many different types of formulations, but the main player here is a progesterone mimic that keeps during the month levels high so that hypothalamus is suppressed. Therefore, no luteinizing hormone, no follicle stimulating hormone, and therefore no ovulation. Okay? All right. Okay. Ending this, I'd like to go into what determines male and female. Okay? Sex determination. I'm not going to go through all embryonic development, that you can have that in a different course, but here talk about sexual development. There are several levels for determining during development, embryological development, whether a fetus is going to become a male or a female. So there's different levels. One level is at the level of the chromosomes, and here we refer to what is called genetic sex. So the genetic sex of the individual does not necessarily correspond with the phenotypic sex of the individual. Usually it does, but sometimes it doesn't, all right? So the genetic sex is referring to whether the embryo has XX, 2X chromosomes, or does it have an X and a Y, okay? Now, there's another level of sex determination called the gonadosex. Now, in the embryo, when the gonads are first formed, they are indeterminate. They are neither testes nor are they ovaries, and they are internal. Okay, they're internal in the development of fetus. And however, due to expression of a gene on the Y chromosome, or the absence of that gene, the gonadal sex later on will become either female ovaries or testes. Okay, And so, in the embryo, about week 6, the gonads are formed, but they are indeterminate. Can't really tell what they are. And however, there are 2 systems of internal tubes that are present. Okay, this is tubes that will become the internal reproductive system. One set of tubes is called Wolffian ducts, and if they develop, that will form the internal male reproductive system. So the vas deferentia and so forth. There's another pair that are there, and both of these are always produced in the fetus about this day, about this week. These are called Mullerian ducts. And if they develop, then this becomes the internal reproductive system of the female. Okay? So the uterus, the oviducts, and so forth. All right, so both of them are there. This is early, and we call that bipotential because either one of them can develop, but which one depends on other things that happen. And this is largely determined in most mammals. Most mammals. You get down to monotremes, however, like duck-billed platypus, their sex determination is really, really different, weird. But nonetheless, in most mammals, the Y chromosome is very, very small, and it doesn't carry much information whatsoever. It has a few genes that affect the spermatogenesis, but what's really important is this gene right here, SRY. In contrast to the X chromosome, which has thousands of genes, this here has very few, the Y chromosome. However, this is all that is needed. So SRY, when it starts being expressed, and so this is not on an X chromosome. So males will have only one copy of it, females will have none, all right? So this here, when it starts, the gene is transcribed, translated, the protein is a chemical signal, okay? And it will bind to receptors, okay? You have to have receptor cells on the undifferentiated gonad, and it leads to those cells, some of those cells developing into Sertoli cells, furthermore, into seminiferous tubules. In other words, the presence of SRY, the protein SRY. So we're talking about 2 things here, the gene SRY and the gene product, protein SRY, all right? That code comes from that. The presence of the SRY protein will induce the gonads to become testes, okay? Because ovaries don't have Sertoli cells, they don't have seminiferous tubules, all right? So, furthermore, it's the Sertoli cells will produce another hormone called anti-Mullerian hormone, or AMH, anti-Mullerian hormone. So specifically, our SRY induces the gonad to become a testes, which has Leydig cells, Sertoli cells. Sertoli cells produce anti-Mullerian hormone. Anti-Mullerian hormone binds to receptors on the Mullerian ducts, which normally would develop as female, and it leads to their degradation. They break down, they're destroyed, they're gone, okay? So this destroys any potential for the system to develop as female. Okay, now Leydig cells are also in the gonad, okay, in the testes. They start producing testosterone. Okay, so both of these cells, Leydig cells, Sertoli cells, are hormone-producing cells. And so that's when I ask for on an exam or quiz the source of these hormones. You really have to go to Leydig cells and Sertoli cells. Okay, so this starts producing testosterone, and this testosterone will bind to receptors on the cells in the Wolffian ducts and induce them to develop. Okay, this chain reaction of things that happen here. Let me run through it again. If there's a presence of a Y chromosome, it will produce SRY protein. That induces the undifferentiated gonad to become testes. which contains Sertoli cells and Leydig cells, both of which produce their own hormones. Sertoli cells produce anti-Müllerian hormone, which will bind to the Müllerian ducts, leading to their degradation. Müllerian ducts, if left alone, will become the female reproductive system. The Leydig cells producing testosterone, that induces the Wolffian tubes to develop. And therefore, they become the internal male reproductive system. Now think about what's going on here. The default condition without the presence of SRY is for the system to become female. The Müllerian tubes don't need any stimulus to develop. They need a stimulus to be destroyed. The Wolffian tubes, they won't develop unless they're stimulated by testosterone. They require stimulation to develop. So if you didn't have any SRY, the gonad automatically becomes an ovary. And there's no anti-Müllerian hormone, therefore these will develop. There's not enough testosterone to stimulate the Wolffian tubes, and they will decay. They automatically will break down. So in mammals, the default condition is to become female. And only if you have the Y chromosome does it make it different. Okay, now one form of testosterone, dihydrotestosterone. This is another form of testosterone. This is necessary to determine what is called the phenotypic sex. Phenotypic sex is the external genitalia. What are they going to look like? All right, so it's necessary for the presence of dihydrotestosterone is necessary for the external features to become male. In the absence of this, by default, they become female. Okay, female is default system. So if we have this, then it will lead to the production of prostate, and the external genitalia will become male. The gonads will sink out of the body into a scrotum. Something that had the potential to either become a penis or a torus will develop as a penis. Okay, only with that hormone. Okay, if it's not there, it won't happen. Otherwise, the external genitalia will develop as female. Once again, and this is here, the presence of this is what determines the phenotypic sex. Phenotypic sex is what does it look like on the outside. Okay, genitalia. All right? Okay, so I already sort of talked about female, but let's just run through the logic here of the female, okay? You have time, I have time, don't worry. All right, so there's no SRY gene, okay, because there's no Y chromosome. And therefore, there's other types of transcription factors, but we're not going to talk about that. So just automatically, without that SRY protein, the Müllerian ducts will develop into the internal system, yes.
So, is there any, have there been any cases where the SRY gene will not turn on the Y?
Oh, yeah, I'll get there.
Okay.
I'll get there, yeah. Probably not today, probably next week I'll talk about that, yeah.
Could you please go back to the previous slide?
Previous slide? Got it? Okay. All right, so there's no SRY. So, as I said, by default, Müllerian ducts will differentiate. And there's no testosterone, there are low levels of testosterone, not as high as in males because the female system isn't going to do that. So, also what's up here is that the gonads will automatically become ovaries. They'll automatically become ovaries. They do not produce high levels of testosterone. Therefore, anti-Müllerian hormone is not produced, okay? That is necessary for the Müllerian system to break down, okay? But we don't have it. Therefore, that Müllerian tube system will develop. The gonads becomes ovaries. And the Wolffian ducts, in order for them to develop into the male system, they require testosterone. There's not enough testosterone, therefore they degrade. Okay, so that now it's just the female. And the androgen levels are not high, especially of dihydrotestosterone, which is necessary for the external genitalia to become male, and therefore the external genitalia will develop into, instead of a scrotum, labia majora, labia minora, instead of a penis, clitoris, and so forth. It becomes female in the absence of dihydrotestosterone. Okay, I might have enough time just for this one last story. Androgen insensitivity syndrome. Okay, there's several different syndromes in which an XY individual can be phenotypically female. Okay, one of them is this. This is a gene, okay, androgen, this here codes for an androgen receptor. Okay, androgen receptor for testosterone. Okay, testosterone has to have a receptor, or else there's no interaction there whatsoever. So the gene, however, for the androgen receptor is on the X chromosome. Okay? Now, if it mutates in a female to the point that the androgen receptor doesn't recognize testosterone anymore, that's in a female not a big deal because they have 2 copies. Males, how many copies would they have?
Just one.
1. And if it's mutated, then it leads to this syndrome. The cells, although they express the protein, the shape is incorrect, and testosterone doesn't do any good. Testosterone doesn't do any good. So if we have an XY individual, they do produce this SRY, and the gonads do develop as testes, and the testes start producing anti-Müllerian hormone, testosterone. With anti-Müllerian hormone, the Müllerian ducts are going to be destroyed, right? Okay? The Müllerian ducts are going to be destroyed, but we have a faulty androgen receptor. The Wolffian ducts don't respond to it, and therefore the Wolffian ducts degrade too. Both Müllerian and Wolffian ducts. So there's no internal sexual system, reproductive system. It's not there. There's gonads, testes, but they don't descend. And also, so it still produces this. So the Müllerian ducts degrade. So there's no functional internal genitalia. Furthermore, the external genitalia will develop as female, even though it's an XY individual. Now, why does that happen? Because the androgen receptor does not respond to dihydrotestosterone. And by default, without that hormone, it'll develop female characters. Okay, all right, so this is where I'm gonna stop. And I just have a little bit more of this lecture before I go into the nervous system. Monday, it's still, the lab is related to the nervous system, but my not talking about what I'm gonna talk about plays no role. Okay, you'll be just fine. Okay, yes, sir?
For the quiz on Monday, can you just let us know, like,
Yeah, it's all of the previous lecture. That is all the hormones, but not the kidney. Not the kidney, even though there was a little bit, I started on Monday, I covered a little bit of the kidney. We'll make it simple. It'll be all the hormones, everything up here. Okay? All right, yes.
are individuals whose genetic sex and phenotypic sex don't match still fertile?
Now, this is still regarded, they don't match, it's due to, because it's